Welcome to my lesson on food tests. Most food tests are qualitative as they are based on changing colour. This tells you whether the biological molecule you are testing for is present or not, but it doesn't tell you how much of that molecule is there or in what concentration. Quantitative results are measurable results such as length, light intensity or time. The Benedict's test for simple sugars, which is the first one I will describe, is semi-quantitative, as there are various resulting colours depending on the concentration of the simple sugar, but it only indicates the concentration of sugar solution. It does not give a figure. Foods in liquid form can be used straight away in these tests. Just remember, if they are coloured solutions, this can affect the resulting colour. Solid foods need to be prepared for most of these tests, although they may be used in solid form in the starch test. To prepare the solid foods, first need to be ground up using a pestle and mortar, and water should be added. After thoroughly grinding the food in the water, it needs to be filtered using filter paper in a funnel and collecting the filtrate in a test tube or boiling tube. It is now ready to test. The test for simple sugars involves adding Benedict's reagent. Add equal volumes of test solution and Benedict's reagent to a boiling tube, then place into a water bath at 80 degrees C and leave for 5 minutes. If simple sugars are present, the blue solution goes through various colour changes, and if a high concentration of simple sugar is present, it will result in a brick red precipitate forming. If a moderate concentration is present, it goes yellow, and a low concentration results in a green colour. The test for starch involves adding iodine solution. Add a few drops of orange iodine solution to the test solution. If it goes blue-black in colour, starch is present. If it stays orange, then no starch is present. The test for protein involves adding Biuret reagent. Add 10 drops of blue Biuret reagent, which is a mixture of sodium hydroxide and copper sulfate. Stir or swirl for 30 seconds. And if a protein is present, a lilac colour develops. If no protein is present, the colour stays blue. The test for lipid involves 2 steps. First, if using liquid food, add equal volumes of the test solution and absolute ethanol, which is a clear colourless liquid. If it is solid food that you are using, grind up the food and add 2 spatulas of it to 2 millilitres of alcohol. Now shake vigorously for 30 seconds and allow any solid food to then settle. Then pour this gently onto cold water. If lipid is present, a milky white emulsion develops at the top of the tube. If no lipid is present, then no emulsion forms. And that brings me to the end of my lesson on food tests. If you found it useful, please click on the like. And if you want to see more of my lessons, why not subscribe?